Really happy to be able to do this virtual experience and being able to be sharing our new data with all of you. And I'm really thankful, Vera, for this opportunity. So as the title is suggesting, the idea here is to show a major role for histone modifications in alternative splicing. And in order to get there, little by little, I would like to start breaking some dogmas. Some of them are pretty obvious for you, some others are not that obvious. First, so we're going to go little by little. The first one, pretty obvious, I think, is that not so far away, not so long time ago, originally we thought that things were pretty simplistic, one gene, one RNA, one protein. But reality is that in the 2000s, with the high throughput sequencing era, we realized that there are barely 25,000 genes, but almost 20-fold more proteins. And the only way of understanding how come we can have so few genes and so many different proteins is thanks to alternative splicing. With this alternative splicing, different modules, coding modules, can be shuffled. You can have some exons included and some exons excluded. You can have even some introns retained, or pieces of exon or pieces of introns included and excluded. And like this, you can have different mature RNAs, different coded sequences that are going to be, by translation, going to give rise to different proteins with different domains, different functions. Thanks to this, first of all, we can increase the protein diversity and the complexity of the same genome. And we can see this in evolution. The more complex the organism gets, the more alternative splicing there is. It also gives a huge plasticity and adaptability, because you can gain easily new functions with this mechanism. And actually, we're realizing now that most of the genes have some alternative splicing, and that it's involved in almost every single process in the cell. And you name it. You can go from cell proliferation to differentiation to sex determination in flies, memory in flies, and pluripotency. And the fact is that when you're talking about something so general, so prevalent, and so important, you are talking about diseases. And those are examples of diseases in which each of them, alternative splicing is playing a major role. In cystic fibrosis, progeria, and muscular dystrophy, it's all about one splicing gene. In neuronal diseases and cancer, it's more about several alternative spliced genes, but in all cases, switching one of the splicing events can have a very important impact in the disease. And in muscular dystrophy is one of the best examples, because it's the first disease in which there is a drug. A drug has been accepted and it's administered to the kids, in which by changing the splicing of SMN, you can reverse the disease. And it's the first time that an RNA can be targeted with a drug. Second dogma that I would like to break, this is a little bit less obvious, is that originally it was thought that you first transcribe your RNA and then you splice and then you translate it into a protein. Reality is that actually most of the splicing events are co-transcriptionally happening. This means that meanwhile the RNA is transcribed, the splicing machinery is recruited and you have the splicing going on, and splicing is finished before the RNA is fully transcribed. And actually, you cannot have the RNA getting poly-A and getting out of the chromatin if the whole splicing hasn't been done yet. Even more, if we now put all this into a nuclear context, reality is that everything is highly compartmentalized and organized. And we have splicing factors organized in this, what is called splicing speckles. It's like a storage place for the splicing factors, and the splicing factors shuffle to the transcriptional unit and come back. They get recycled. And it is known now that there is an intimate crosstalk between the two. And if something prevents splicing or inhibits splicing, then you cannot have transcription elongation anymore. They really need each other. And even more, I will push it a little bit further. And I just want to highlight that there's so far only one paper suggesting such a thing. It was published recently in Nature Communications, but it's a very interesting and provocative idea. And is that now it's pretty much accepted that different loci can come together, especially can come together in the nucleus because of transcription. If they're sharing transcriptional regulators or units, they can come together for a more organized and efficient transcription. And they're called transcription factories. And it turns to that in muscular, during cardiomyocytes differentiation, this group realized that there is a splicing factor called RBM20 that could also be responsible of this coming together of the loci into what they call splicing factories. And if they knock down the splicing regulator, those loci will come apart, suggesting that it's not only about transcriptional units, but also co-regulated splicing units. Then the third dogma I would like to break before getting to the data is the realization as well, recently, thanks to single-cell transcriptomics and particularly splicing analysis, which is not straightforward at all, that splicing is not a black and white thing. Alternative splicing, sorry, is not black and white. It's true that most of the alternative splice events will be either completely excluded or completely included, but you have a 20% of the splicing events that will be multi or bimodal, which means that you will have splicing events shared, present in the same cell, or then there will be some cells with one splicing event and cells with the other, or then as well, and that will be one of the most prevalent cases, in which you have cell-cell and transcript variability. And even more, a work in which they did single-cell splicing analysis during differentiation of induced pluripotent stem cells into motor neurons, they realized that it's around 20% of these alternatively spliced events that are actually changing during this process. And those changes will never be black and white, from highly excluded to highly included, but also going through this bimodal, multimodal, suggesting that we're having shades of gray, and that we have this complex regulation of splicing that goes from cell to cell, or even transcript to transcript in the same cell, raising the question how this can be so tightly regulated. And that's where chromatin comes into the picture. And in the late 90s, early 2000s, an increasing list of evidences showed that transcriptional regulators, chromatin binding proteins, nucleosome remodelers, the nucleosome positioning, DNA methylation, histone modifications, all of them have been shown in a way or the other to be impacting the final splicing outcome. And most of them have been shown or suggested to do so, but by modulating RNA polymerase II kinetics. And briefly, what this means is that, remember, this is co-transcriptional. And so by modulating POL2 kinetics, you're modulating the window of time of opportunity for the splicing machinery to be recruited to weak splice sites. So if you slow down the POL2, those weak splice sites will have more time, we're talking about nanoseconds, than the more stronger splice sites coming after. And like this, the exon, the splicing machinery will have the opportunity to be recruited. And by this, I mean both activators and repressors. This doesn't mean that slowing down POL2 increases inclusion. It just means that you have the splicing factors that can be more recruited and can lead to inclusion or exclusion. So it's pretty complicated. Reality is that during my postdoc, and other labs did too, is that those histone modifications can have a more direct role in alternative splicing, not only by modulating the kinetics of POL2, but also by directly recruiting the splicing regulators through chromatin binding proteins. And here, what we found is that there can be exons enriched in specific histone marks, and thanks to specific chromatin binding proteins that will recognize those histone marks, there will be a protein-protein interaction with the splicing factor that will be recruited to its splice site. And as you can see here, you can have the same histone modification recruiting different splicing factors via these chromatin adapter complexes, or same splicing factor recruited by different histone marks, really increasing the complexity and specificity of the whole reaction. Now, it's not only about which splice sites you have, but also which histone marks are enriched, locally enriched around this area. So the questions that we got in the lab when starting in 2013 were quite a few. Some of them were managed to answer, some of them are still ongoing. And basically, the first very question from everybody working on the chromatin and splicing field is, which is the global impact of these histone modifications. How many exons are actually regulated by these histone marks, and what do these exons have in common? Why those exons are not others? Another question that raises as well is, who regulates those histone modifications, those splicing-associated histone marks? Who determines if you have the green one or the red one? Another very important question is, are these histone modifications modulators, fine-tuners, or are they actually playing an active role? Are they drivers, inducers of those splicing changes? And finally, the mechanisms. We know so far they can regulate Pol2 kinetics that do have these chromatin adapter complexes, but are there other mechanisms involved, and which ones? And so in order to answer those questions in the lab, we have three main axes. The role of non-coding RNAs in splicing, 3D chromatin organization in splicing, and chromatin histone modifications in splicing. And all of us use the same cellular model system to answer those questions. And this is the epithelial-to-mesenchymal transition. And why this model system? First of all, because it's very biologically important and physiologically meaningful, because this reprogramming of an epithelial cell into a mesenchymal cell happens naturally during early development or in wound healing. But unfortunately, cancer cells takes advantage of this for gaining migratory and invasive phenotypes, and therefore being able to do a metastasis or resist to treatments. Moreover, this reprogramming is very dynamic. In one day, we start observing the first changes in chromatin and splicing, and by 5-7 days, it's done. The cell has completely changed its morphology and its phenotype. It's inducible and reversible, that goes forward into this dynamism, and also it's completely splicing dependent. There is a list of splicing events that change during EMT, and by just touching at one of the splicing events, you can prevent EMT or you can induce EMT. So splicing is a major regulator. So in this talk, I'm not going to be talking about the non-coding RNA branch, because that has been already published in 2015. I'm not going to be talking about the 3D chromatin organization, because this is ongoing. Hopefully soon I will be able, but not yet. I want to talk about the chromatin axe, and I'm going to be talking about two stories. One, our genome-wide approach, led by Enedit Aguirre, a very brilliant computational biologist that is now in the Karolinska Institute. And then a more gene-specific approach in order to understand the role of histone marks in splicing. And that has been done by two amazing researchers, Yaiza Núñez Álvarez and Alexandre Segeb, who I'm really very lucky to have in the lab. So first thing, our genome-wide approach. So Enedit wanted to address the global impact of these histone modifications and which are the histone marks playing a role in alternative splicing and why some exons and not others. And to do so, she wanted to go big and she wanted to go broad. And so she took advantage of available transcriptomics and epigenomics data from the ENCODE and the Roadmap Epigenomics project. And so she chose the two cell lines, human embryonic stem cells and mRNA-90 fibroblasts, in which there was the most extensive data sets of CHIP6 and MEDIP6. MEDIP6 is DNA methylation. And different from the rest of papers, because there is a bunch of groups that have tried to do such a thing already, she looked at alternatively spliced exons, but classified them into four categories, from highly included to highly excluded, but also paying attention to the mid-included and the mid-excluded. I remind you that exons are not black and white, it's not fully included or excluded. You have shades of gray, and she wanted to address what happens at these shades of gray. And the strategy was to first map those histone modifications at those alternatively spliced exons, and then look using random forest classifiers, machine learning classifiers, which were the histone modifications, which enrichment in these alternatively spliced exons were informative to classify them into these four splicing categories. And she did this in mRNA-90 cells, and she did this in H1 embryonic stem cells. Obviously, the ranks will be different because there are different cell types, different qualities of the CHIP6, but she took overlapping histone marks And she looked at differential enrichment of these histone modifications at those exons. And unfortunately, most of those alternatively spliced exons were not really very clearly enriched in one category on the other, which was not very surprising because that was a little bit the conclusion of the other works. But Fernandez didn't surrender into that, and she said, wait a minute, we know that histone marks play a combinatorial role in transcription. What about in splicing? And so she started looking at co-enrichment of two histone marks, and she did two histone marks because that's the first step, that's the easy one. Co-enrichment of two histone marks at an alternatively spliced exon by category, and also by position at the exon upstream or downstream. And she will only keep the combination of histone modifications that will be enriched only at a specific category and at a specific position. And what she found is that effectively there will be 7 different splicing-associated chromatin signatures that we call SACS. Those SACS will be differentially enriching 31% of all the alternatively spliced exons expressed in embryonic stem cells. We will see that those SACS will be highly combinatorial and position dependent. And what I mean by that is that you will have, depending if you're included or excluded, more than one combination. And as well, depending which combination, you could go with inclusion or exclusion. We will have different combinations for the included or excluded. So we will be not only one combination only with inclusion and one combination with exclusion, but we could have more than one. We will also have specific combinations for the mid-included and mid-excluded. That goes well with the idea that we have shades of gray, and chromatin will play a role in these shades of gray. And also, it will be important that it's all about this combination, because you would have the same histone mark enriched at either included or excluded, and it's all about which other histone mark is co-enriched with, that will be differentially enriched with more inclusion or exclusion. Same-wise, it will be highly position dependent, since the same combination of chromatin modifications will be differentially marked excluded or included, depending if they're enriched at the body of the exon or more at the end. And this has also been observed by a different lab, the GIL-AS lab, and we observed that there were differences in RNA pol 2 enrichment at these 2 types of exons. So this shift of this chromatin, of this SACS, will impact RNA polymerase 2 posing and elongation rate. Another thing we realized as well is that depending on if the exon will be enriched in one SACS or the other, those exons will have different exon sizes. Most of them will be smaller than controls and constitutive, and some of them will be extremely large. The weakness in splices will also be different depending on the SACS. Those SACS will have weaker splices than those SACS. We also observed, and that was pretty surprising, that depending on the SACS you will be enriched, those alternatively spliced genes will have different gene ontology terms associated and not in common, suggesting that SACS will be co-regulating functionally related alternatively spliced events. And finally, it will not be about functionality, but also about regulation. Those exons co-enriched by a specific SACS will be also enriched in specific RNA motifs. And depending on the SACS you will have, you will be more enriched in one splicing motif or splicing factor or the other, suggesting a coordination, sharing of specific splicing factors depending on this SACS. Importantly, when looking whether those SACS will be conserved between cell lines, what we did is to now work on the same exons. So we're not working in different SACS marked exons, but in the same SACS4 exons in here. And we will look what happened at these exons in other human cells, other cell types, and look at the enrichment of these histone marks in those cell types. And what we observe is that these exons, when they will be kept excluded, they will also keep the enrichment if these SACS marks. However, when they will switch to inclusion, to another splicing category, they will not be specifically enriched in this mark. So changing of splicing will change of enrichment of these SACS. And that would be more obvious in this example, in which in embryonic stem cells, those exons are enriched in SACS3, they are mid-included. When kept mid-included, they will be keeping this SACS enrichment, but when they will be switched to exclusion, there will be change of SACS. They will not be enriched in the SACS3 marks, but they will be enriched in the SACS4 marks, in the blue marks, suggesting that a change in splicing will correlate with a change in enrichment of these histone modifications. And even more, I talked about RNA motifs before, and we saw that it's not only about changing or maintaining a specific chromatin combination, but it's also about maintaining this enrichment of the splicing factor. And here we took advantage of available ClipSeq data. It's basically RNA immunoprecipitation of a specific protein. And what we observed is that the exons enriched in SACS4, when kept excluded in the other cell types, they will keep this enrichment, this SACS4 enrichment, but also they will keep the binding of HRNPK. And when excluded, they will lose the SACS enrichment and the HRNPK binding. So in conclusion of this first part, what we observed is that actually a third of all the alternatively spliced exons expressed in a specific cell type will be potentially differentially marked by a combination of histone modifications in a highly positioned specific way, what we call SACS, suggesting that there could be a histone code in the splicing code. Also, we observe that those SACS are conserved between cell lines, which doesn't exclude that there could be cell-specific SACS or even disease-specific SACS, and that would be very important for targeting those events. And then finally, the biggest question that raises is that all those are correlations. We still do not know if those histone modifications, these chromatin signatures, play an active role in alternative splicing. Are they a cause or an effect? Is this just a correlation, or are they actually functionally relevant? And in order to address this functionally relevance, we decided to move into a functionally relevant model system in which we could modulate, in which we could change those histone modifications, and that would be the epithelial-to-mesenchymal transition. And now we moved into gene-specific approaches. No more genome-wides. Now we are addressing specifically a gene, and we're manipulating that gene. And so some background in EMT and splicing in order to understand what we're going to be doing. So as I said before, EMT is an inducible reprogramming system, and obviously it cannot be simple. It's not about an epithelial cell that turns into mesenchymal period, but it is progressive. We have different intermediate states, and there's lots of cell-cell variability. All those different cell states are changing in time, and it's not entirely known yet how many different cells we have and how they really change. It's ongoing, but it is important to know this, because at the splicing levels is exactly what we observe. We have different dynamics. We have also a progressive change in alternative splicing, with some splicing events changing very quickly in 12 hours, and some other splicing events changing a little bit later in 24 hours, not in 12. Rarely we have absolute changes in these inclusion levels, and this means going from 0% inclusion to 100% inclusion. In this gene, we do have a very quick and absolute change. Most of them are really very progressive, and they go from 0 to 30% inclusion or 30% to 0. And 30% inclusion means that either 30% of the cells have inclusion, or either 30% of the transcripts have inclusion. So we're talking about the cell-cell transcript variability, and therefore probably chromatin modifications involved in this regulation. So in order to address this, we decided to focus everything into two well-known alternatively spliced genes, very important for EMT. This is FGF receptor 2 and CTND1. FGF receptor 2 is a receptor, transmembrane receptor. It is essential for the reprogramming. because depending on which exon it is included, it changes completely the capacity of the receptor to recognize one ligand or the other, and therefore the secondary cascades are completely different, from highly differentiated epithelial cells to not so differentiated stem cell kind phenotype, migratory invasive phenotypes gain. And CTND1 is it's an associated protein to cell-cell adhesion, to E-cadherin, and a change from the alternative splicing from epithelial to mesenchymal changes the capacity of interaction with E-cadherin and changes completely the cell-cell adhesion. And you have the apical, the epithelial polarity. And so by gaining the mesenchymal isoform, those cells don't need to be in cell-cell contact, and they gain migratory and invasive phenotypes. So these two are very much involved in metastasis. So the strategy we use here is the first approach, do correlations, but with with time courses and around the genes. Those are chips. We are focusing into the alternatively spliced exons in CTND1 here, and also looking upstream and downstream along the gene. Here are the histone modifications we're looking at, and those are normalized. We're always normalizing the chip levels by background levels. Like this is more comparable between EMTs. And here we're comparing untreated epithelial cells and in orange, EMT-induced cells. And we're first doing this in T7, where the changes in alternative splicing are complete, EMT is complete. And what we observed is that during EMT, at this particular exon and only at this exon, we have a significant increase in trimethyl 27 marks, whereas there is a decrease in acetylation, in 27 acetylation, and a decrease in mono K4. What is really interesting is that when we looked at 6 hours post-induction of EMT, no changes in splicing are still detectable. We still observe these changes in histone modifications. The trimethyl K27 was enriched and K27 acetyl was decreased before any change in splicing could be observed. However, mono K4 was not changed yet. Mono K4 seemed to be changing later in time. And all those changes were reversible. When we had we did the reverse mesenchymal to epithelial transition, which means that splicing is reversed back to epithelia, those changes in chromatin will also be reversed. So we have dynamic, highly dynamic changes in chromatin that precede the dynamic changes in splicing, suggesting a functional role. And it's important to note that was not only true for CTND1, but we also observed those correlations in histone modifications and splicing in other important key genes for EMT, such as FGRECEP2, SLK script, and TCL7. And also it's important to highlight that it was not always a gain of K27 trimethyl or a loss of K27 acetyl. Each of them will behave differently, suggesting, as I said, a more, not as something very specific so for CTND1, but a more global role for those marks in alternative splicing during EMT. So in order to address now this actual role of these histone marks, we wanted to do something very specific. We really wanted to address the role of these histone modifications at this exon and only at this exon. No pleiotropic effects in the whole epigenome. And the only way we can do such a thing, we can be so localized and specific, is taking advantage of the CRISPR-Cas9 epigenetic editing system. This system consists on the use of the DNA targeting Cas9 protein that is mutated. It has a mutation that makes it unable to cut the DNA. So it is recruited but cannot cut. And then you can fuse to the Cas9 any catalytic domain of interest. In our case, histone writers. And by targeting this fused Cas9, you can modify the histone mark of interest in a highly localized way. And we know that it has a 500 base pair range of modification, and then address what happens to the splicing and what happens to the EMT. And since what we observe is that the histone marks changing were K27 trimethyl and K27 acetyl, we fused to the dCAS9 the catalytic domains of the most well-known, the more prevalent histone modifying histone writers for each of these marks, with ECH2 increasing trimethyl K27, UTX demethylating K27 trimethyl, P300 acetylating K27 acetyl, and SID4X deacetylating K27 acetyl. And the first thing we did was to verify that we were actually changing the chromatin. So in here is, I'm only showing you ctND1 for simplicity, but we did the same in FG receptor 2. We always target more than one gRNA to increase efficiency. We target the gRNAs to the exon. So this is exon 2, the exon of interest. And as a control, we also target exon 20, another unrelated exon, to make sure that this is specific to the targeted exon, not because of off-target effects or effects of just overexpressing the catalytic domain. So those are all chips, and the first thing we did to verify that the dCAS9 is properly targeted to the ctND1 exon 2 in every single combination. And then we looked at the levels of trimethyl K27 and acetyl. And what we observe is that when using non-targeting gRNAs or gRNAs targeting exon 20, no changes in exon 2 trimethyl 27 levels. When targeting an empty dCAS9, no catalytic domain, no effect. And we only saw effect in the trimethyl K27 when actually targeting the modifiers of K27. VSET, that is like the viral set domain for trimethyl K27, is like ECH2, increased in a 5-fold the methylation levels of trimethyl 27, whereas UTX reduced it very slightly, not efficient at all. Surprisingly, an increase in acetylation also impacted trimethyl 27. As you can see, P300 is actually reducing trimethyl 27 much better than the demethylase. And we can see here that P300 is efficiently increasing the acetylation levels. SID4X is decreasing it. And again, we also saw an impact of the trimethyl catalytic domains, highlighting that these two marks are kind of mutually exclusive. Impacting one has an effect on the other one, but that was already published. And as well, showing that by recruiting those dCAS9s, we can recapitulate pretty well what's going on during the EMT. So now, what happens at the splicing level? So here, just a reminder, during EMT, exon 2 in ctND1 increases, gradually increases. And this increase in exon inclusion goes with an increase in trimethyl 27 at the exon 2 and a decrease in acetylation. So an increase in trimethyl 27 should increase splicing levels, whereas a decrease in acetylation should also increase the splicing levels. And that's exactly what we observed. SID4X deacetylates K27 acetyl, and we have an increase in the splicing levels, whereas VSET and ECH2 that increases K27 trimethyl increase the levels of the splicing, increase the splicing levels. And P300 and UTX, P300 will increase acetylation and UTX will decrease K27, had no impact because that would be an epithelial conformation, suggesting or proving that those histone modifications are sufficient, can induce a change in splicing all by themselves, because the only thing we're doing is changing a histone mark in a specific exon, raising the question whether those marks not only are sufficient, but also necessary for those changes in splicing during EMT. And for addressing this question, now we had to turn into drugs, unfortunately, because we need something highly dynamic. And the whole idea here is that as soon as we induce EMT, we're using a drug, an HDAC inhibitor, that will prevent this decrease in acetylation, will maintain this increase in acetylation, and therefore will not allow neither this high increase in trimethyl K27. With this inhibitor, we will maintain the epithelial chromatin state while the EMT will continue. First thing and because those inhibitors are pretty toxic, we managed to go until T7, but actually all our effects were mostly observed in T1 because of this toxicity. And importantly, this EMT was not affected. We could have a normal EMT as expected. But when we looked at the inclusion levels of 2 exons in which you need this decrease in acetyl levels and increase in trimethyl levels of K27, the inhibitor did not permit this change in inclusion as expected in normal conditions. Whereas other exons not sensitive to K27 marks will not have any problem in doing properly the switch in alternative splicing during EMT, suggesting that those histone modifications are not only sufficient, but also necessary, and they could be key drivers for the changes in splicing, the dynamic changes in splicing that occur during EMT. Next question was, how are those histone modifications impacting splicing? What are the mechanisms? And for this, Yaitha did a tremendous effort looking at every single splicing factor from literature or any RNA binding motif search analysis that could be playing a role in ctDND1, and she screened it with her bare hands doing shRNAs. And she observed that from all these splicing regulators that potentially could be playing a role in ctDND1 splicing, PTB was majorly the one which recruitment will impair inclusion, since in its absence you will have more inclusion. The next thing which she did was to look at during EMT if it's true that there is a change in PTB recruitment during EMT, and effectively she observed that at exon 2, the exon of interest, during EMT, PTB will not be recruited anymore, and that's why exon 2 gets more included. Whereas in other control exons, there will not be changes. This exon 6 is always excluded, and this exon 20 is always excluded. Importantly, when she did this DCAS9 epigenetic editing in order to increase inclusion of exon 2 by increasing the trimethyl K27 levels, those are epithelial cells. The only thing she's doing is recruiting DCAS9. She observed how PTB was not recruited anymore, suggesting that the way these histone modifications are impacting splicing is via modulation of recruitment of these splicing factors. And finally, the final question, the most important question in all this is, fair enough, these histone modifications impact splicing, but are they impacting splicing in a biologically meaningful way? Are we impacting EMT in all this? And so the first approach was just targeting our DCAS9. We're only showing in here the DCAS9 that when targeted in epithelial cells increase inclusion of ctDND1 of the mesenchymal isoform. And we looked by RT-PCR and FACS, the protein levels, at markers, well-known markers of EMT. And we have a slight reduction in epithelial markers, a slight increase in mesenchymal markers, and even an impact on migration when healing. But it's always mild. It's never, not even close to what would you observe during EMT. A little bit disappointed. But then we talk to DCAS9 experts and they say, careful, DCAS9, even though expressed, is not very efficiently targeted to the chromatin. And you should be verifying how many of your cells are actually properly modifying this splicing. And we realized that effectively, thanks to the use of splicing-specific antibodies, around 20% of the infected cells were actually changing the splicing. And so we decided to cell sort for the cells in which they will be actually doing this change in splicing, in which we know that DCAS9 was working. And what we observe is that initially, using this splicing-specific, this antibody that only recognizes the mesenchymal isoform. So in epithelial cells, there's barely 8% of the cells that have this mesenchymal splicing isoform expressed. This is like a known leakiness of the system. When targeting a mutant VSET, which means that doesn't have any catalytic capacity, you don't really change this percentage. During EMT, you have a 43% of the cells that increase this expression of the mesenchymal isoform. And by targeting VSET, you increase to 31%, close to what we observed during EMT. And by now looking at the effects on exon 2 inclusion, what is really, we will observe that now we can totally recapitulate the splicing changes. Now we're changing the splicing exactly at the same levels as EMT. Before we were around 0.2, now we're 0.3. And regarding EMT markers, now we could totally recapitulate EMT. We can really see by RT-PCR and by protein levels by FACS that we have a complete depletion of the epithelial markers and a complete increase in the mesenchymal. And we could also have the same gain in phenotype, both at the bidirectional migration as an invasion assays, suggesting by inducing those localized changes in chromatin, we could change the splicing to an extent that it would be sufficient to recapitulate the EMT. And so, as a second conclusion and proposed final model, we're showing that a change in histone marks just at the exon of interest is sufficient and necessary to induce a change in splicing. Those changes in splicing can recapitulate EMT, so the splicing-associated chromatin modifications are biologically relevant, and this regulation is highly dynamic, suggesting that those histone marks could be drivers that are biologically meaningful, and that those exons could be poised, that those exons could be already changed in a first place, allowing this highly dynamism when necessary in specific cases, such as in this reprogramming or in response to specific stimuli. And so in perspectives, and to be finished, I would like to highlight, to put all this together and suggest or think that chromatins plays a role in coordinating functionally and regularly related splicing networks, and that the dynamic nature of these epigenetic modifications could be a huge advantage in the cell for these rapid reversible splicing responses, and also for the cell-cell and transcript heterogeneity variability. And now the whole lab is moving towards these concepts, so going single cell and also single locus, because we want to look at what happens specifically at this transcript-specific changes in splicing, and always with the idea of impacting cancer, of going physiological. And we are striking important collaborations with oncologists in Montpellier in order to address this. And so to be finished, this is really one of the most important slides. I want to acknowledge and thank all the members of the lab, without whom this work would not be possible, present and in the past, our collaborators, and of course, our funding agencies. And thank you for listening to our story. Well, the visions around you.